Wonderful. So we are excited to open our Bar-Ilan University Vision Science Seminar today, and we are delighted and honored to have with us in our seminar Professor Gabriel Kreiman from Harvard. Gabriel did his BSc in physical chemistry in Argentina at the University of Buenos Aires. He then continued to do an MSc at Caltech in computational and neural systems under the supervision of Professor Christoph Koch, and then also a PhD in biology under Professor Koch's supervision. He then became a Whiteman Science Fellow and McGovern Institute Fellow at MIT's Department of Brain and Cognitive Science and Computation and Systems Biology Initiative, working in Professor Tommaso Poggio's lab. Today, he's a professor at Harvard Medical School and the Children's Hospital in Boston, where he leads the Kreiman Lab and is part of the Harvard Center for Brain Science and Harvard University Center for Minds, Brain, and Machines. Throughout his career, he won multiple prestigious awards, including NIH New Innovator Award, NSF Career Award, Society for Neuroscience Career Development Award, and many more. And I want to point out what I found fascinating is also the Metropolitan Chess Champion Award in Buenos Aires for kids under 15. He has more than 19,000 citations, and his work is published in top leading journals as Nature, Science, Neuron, and many other top leading journals. He also published multiple book chapters and reviews, has been invited to dozens of invited talks across the globe, has and is mentoring many successful and talented students and postdocs, and also several high school students, and there is much more to say. Importantly, his research encompasses vision from multiple angles and levels in both living and machine vision, looking into many dimensions of the visual experience. He combines electrophysiological measures, computational and modelistic approaches, deep neural nets, naturalistic and lab-based settings, eye tracking investigation in his research, looking into object recognition, memory, attention, cognition, and these are just naming a few of the many topics his lab investigates. So without further ado, I'd like to welcome you, Gabriel, to our seminar, and we'd love to hear about your research.
Thank you very much for this very kind introduction. Let me share my screen. There it is. Can everybody see my screen?
Yes.
Okay, perfect. So again, thank you very much for the very kind introduction. And I'd like to tell you, I gave Sharon a menu of a couple of options, and she wanted to know more about neurons. So you're stuck with the question of what do visual neurons want? I'll try to define that question and tell you why I think it's important and interesting to discuss this. And I think this can change how we think about neural coding for visual processing in cortex. All the work that I'm going to talk about today was done jointly with a number of very talented students and colleagues who are all mentioned here: Alex Bardon, Till Hartman, March Livingstone, Carlos Ponce, Peter Schade, and Will Shao. Throughout the talk, I will have some of these QR codes. If that works, you can scan them, and that will lead you to a website where you can download talk, papers, data, and code. I told Sharon already, and I apologize beforehand. Unfortunately, I have a thesis defense in 1 hour, so I'll have to leave right at the hour. But please do interrupt me with questions, and if I don't get to answer all your questions, feel free to reach out and send me an email, and I'll be happy to try to address your questions. Okay, so my talk is divided into 5 parts. First, I'd like to define what I mean by what neurons want. Then I'll give you a couple of examples from units in computational models and ask what units in computational models want. I will introduce a new method that was presented by a student, Will Shao, in our lab to try to interrogate tuning preferences of neurons in an unbiased and systematic way. I'll show you how that method works in silico, and then I'll show you how it works in vivo, in neurons, in monkeys. And finally, I will use this method to argue that phase neurons are actually encoding non-semantic features in cortex. Okay, so first, just to start from the very beginning, neurons really do not want anything. What I mean by that is what kind of visual stimulus triggers high firing rates. why are we obsessed with firing rates? Well, this is the main definition of neural coding that people have been using for decades. So if you go back to Hubel and Wiesel, the discovery of orientation tuning means that there are neurons that respond to some oriented bars with higher firing rates than other neurons. If you look at the Nobel Prize winning work of John O'Keefe with place cells in the hippocampus, that means that there are neurons that fire that have higher firing rates in some locations in the environment compared to others. So that's the classical definition of neural coding. Everything I'm going to say will be restricted to flashed visual presentations. It will be restricted to passive viewing. And importantly and critically, we want to be able to study neural coding under realistic experimental conditions, which means that we have finite recording time. And this is critical because the universe of images is enormous. So for example, just to introduce the course of dimensionality and what the problem is about, imagine that we want to present pictures, record the activity of neurons, and figure out what the neuron likes, that is, what kind of pictures elicit high firing rates. Imagine that we have an image that's 100 by 100 pixels. This is a pretty small picture, but just for the sake of argument. And imagine that we forget about color for a second, and each pixel can have 256 shades of gray. How many such pictures are there? So you can do a little bit of math and you can quickly figure out that there are 10 to the 24,000 such images. So even for a very, very small patch of 100 by 100 pixels, it is prohibitive to exhaustively study the entire stimulus space. So if you have an amazing grad student that doesn't drink, doesn't sleep, doesn't eat, doesn't do anything other than record neural activity, and if you show 5 pictures per second, which is typically what we do in many of our experiments, in 5 years, this student could present about 10 to the 9 images. So there is absolutely no way that we can exhaustively examine the entire sampling space. So we need better methods. So what and so if you add color, the number is even higher. That's shown here. So among all the possible images that we could present to a neuron, how do we figure out what the neurons actually respond to? So in the classical world, and this is mostly what we and everybody else has been doing for the last 5 decades or so, one option is to rely on previous studies. So we look at what kind of pictures people have used before, and we use the same pictures because that has worked. For example, we have 5 decades of studying gratings. Another option is to study images based on some intuition. So for example, some people believe that faces are interesting, or angles or curves, or different types of intuitions about what kind of pictures should matter to the nervous system. Another option is to rely on natural image statistics under the conjecture that what happens in the natural world should be represented in the brain. Another option is to use computational models, and I would contend that this has not been used too much in guiding what kind of hypothesis or pictures to present to study neural coding. And the other option is serendipity. And honestly, a lot of the discoveries about neural coding in cortex, including the seminal work of Hubel and Wiesel, was basically a serendipitous discovery by very astute people that were really thinking very carefully about what was going on, but they sort of landed on some of the preferred stimuli almost by chance. So in this way, people have recorded the activity of neurons in the retina and found out about center surround receptive fields, orientation tuning in V1, motion in area MT, neurons that respond to complex shapes, including faces in inferior temporal cortex. So what we really want to do is to try to study in a more systematic way what neurons want. And I'll first start by talking about what neurons want in computational models, and then give you a flavor of the kind of methods that we introduce to study this question in a more systematic way. Why do we want to focus on computational models? Well, in computational models, we can know absolutely everything about every single unit in the model. So I think it's a good test bed to begin to investigate the preferences of neurons in cases where we know by construction, by definition, what the units should respond to. I'm going to go through this somewhat fast, and I hope people are somewhat familiar with neural networks. So here's a neural network that we built that's extremely simple. So we have a picture, we have 3 convolutional layers, and then a fully connected layer at the end that's doing classification. So it's a very, very simple neural network, just to illustrate a couple of points about how units in this network respond to visual stimuli. So we trained this very simple network with pictures from 6 random categories chosen from ImageNet. In this particular example, these are examples of those pictures. So we have pictures of roses and sports cars and fire ants and biological cells and Labrador dogs and so on. This is a t-SNE representation, that is a slow dimensional representation of the activations of the units in the top layer, that is in this classification layer, to all the different pictures that were presented. And you can see that each color represents one of these different categories. So there's some separation, meaning that this network can separate these images relatively well. Nothing too surprising here. Computer vision has excelled in the last several years in many of these pattern classification problems. But what I want to tell you is, what are these units doing? These units are doing classification, and I want to show you how these units are activated by different stimuli. So what I'm showing you here is the first FC unit, so the first unit here, and I'm showing you the activation strength in response to all the 6,000 or so different pictures that were presented. And you can see here that this unit is particularly activated by the first category, that's the red category. And this is good, that it should be so, because this unit is actually classifying, whenever this unit is active, the model thinks that category was presented. So indeed, there is a stronger activation for that, for images from that category. But you can see that it's not all or none. That is, there are other pictures from other categories that also trigger relatively high activation, and there are images from that category that trigger low activation. So this image, even though we know by definition, by construction, that it should respond to the first category, it's still mixed up. It still responds to many of the other images. And the same thing happens for all the other 6 units within this computational model. Furthermore, we can ask the following question. We can take a category that the network was never trained on, that is, the network should not care at all about other categories, such as faces and houses. And I'm choosing faces and houses because this has been extensively studied in neuroscience and cognitive science. So we can present now pictures of faces and houses and ask whether this network can separate those pictures. And lo and behold, it can do so extremely well. So here's an example unit here in the bottom. I'm showing the activation strength as a function of image number. So here we have different pictures of faces, different pictures of houses, and this unit can separate faces and houses, so it has a stronger response to faces and houses. Some may even call this a face unit, even though this unit, we know by construction, is not a face unit. So that is, we can very easily get confused and deluded in the classical experiments that we do in thinking that this is a face neuron, when clearly it's not by construction. Because we know every aspect of the network, we can do what's called deep dreaming. That is, we can ask and basically regress out and propagate back and ask what kind of picture will trigger maximum activation. So here's an example of doing that for the Labrador image unit, meaning that the top unit that responds to Labrador images. So here again is the activation for all the images. This particular unit responds more strongly to pictures of Labradors compared to all other images. But then we can also create synthetic images like this one here. This image is not an ImageNet, and this image here responds way stronger than any image in ImageNet. So this image leads to a much stronger activation for this particular unit. This is another example for this for another unit. In other words, we can create synthetic images that trigger much higher activation than any natural image that was used in the training set. Okay, so to summarize this part, we think it's useful to use models as a test bed, at least just to conceptualize some of the key concepts. Even the output units, which we know by definition are classification units, do not necessarily contain semantic information, and they actually respond to a variety of different shapes. And we can also discover pseudo-selectivity. For example, a model that has never seen a particular category A can have units that discriminate A versus B, just because that particular category A, for example, faces, can correlate with what the neurons or what the units in the network are actually interested in. Okay. Okay, so this is just a proof of principle to show the complexities of studying unit preferences in a case where we can understand everything about the system. So what we wanted to do was come up with an algorithm to probe tuning in a systematic way. So I will show you the algorithm that we called Xdream that was introduced by Will Shao. So what we wanted to do is we wanted to study how do we know that we have truly uncovered what neurons like. So we have a neuron, we're recording the activity of the neuron in response to different pictures. And again, we can present different gratings, different orientations, we can present pictures of faces and houses, we can present natural images, we can do all of these experiments, but at the end of the day, how do we know that that's really what the neuron is tuned to? Perhaps there exist other stimuli that we just haven't tested that can drive the neurons in a way that's much stronger than any of the natural stimuli or any of the gratings or any of the pictures that we have been using so far, in the same way that we have just shown that happens in computational models. So we want to have an algorithm, and the Desiderata for that algorithm is that this algorithm should be able to generate images. These images are hypotheses of neural coding. We want to be able to explore a vast stimulus space. Ideally, the algorithm should be able to explore the entire image stimulus space, which, as I mentioned earlier, is enormous. We want the algorithm to be unbiased, or at least less biased than investigators saying, I'm going to choose and present pictures of chairs or faces or gratings. And we want this to be actually applicable and actually work in real time with the constraints of the neurophysiological recordings that we have at our disposal. Okay, so here's what the algorithm looks like. So this is a closed loop algorithm that basically takes one of these neural networks like the one I just showed you and turns it upside down. And by turning this network upside down, this network, instead of taking pictures and classifying them, this network will take features and will generate images. So this is an image generator similar to what a lot of people have used in GANs, that is in generative adversarial networks. So we will start with noise, and we will be generating images from those codes, from those features that are the input to this image generator. We're going to present those pictures to a monkey. We're going to record the activity of the neuron. We're going to use the activity of the neuron. We're going to let the neuron dictate what it likes. So we're going to use the activity of the neuron to select which images seem to be good, meaning which images trigger high firing rates. And then we're going to use that to combine and select features to go back to the image generator, and this can be run in a loop multiple times with the goal of continuously increasing the firing rate. So the goal here is to start with noise and in a systematic way to try to modify the images that are generated by this image generator here to try to come up with images that trigger very high firing rates in these neurons. So again, the first step and the key step here is to have an image generator. In the classical neural network community, what I showed before, we have a picture, it goes through a network, and we generate a list of features or codes in different layers. And so what we're doing, which is similar to what a lot of people have done in the neural network community, is to turn this upside down, basically. And so we are going to start with features or codes, and we're going to be able to generate images. We tried a lot of these different image generators. In this particular case, this is a famous algorithm called AlexNet that's basically turned upside down to be able to generate images. The starting point is noise. So we're going to generate images that are just pure noise, and then we're going to use those as input to the algorithm, and then try to evolve images from there. So in the first step, before I show you how this works with real neurons, we're going to test this algorithm with actual neural networks. So in the neural networks, again, we know what the units should respond to. So we can use this as a test to assess whether our algorithm is working or not, okay? So first, let me show you cases where we know the absolutely, the global ground truth, that is the global optimum for the unit. So we synthesize a unit where this is the target, meaning that this is the image of all possible images in the world. This is the image that will trigger the highest activation for this unit. And we then run this algorithm starting with noise, and this is what we can generate. It's not exactly identical, but I think it is pretty close. Similarly, here are more examples. So we come up with a couple of random examples, including a picture of a Coke bottle in front of Neptune. And then we start our algorithm from noise without any assumption whatsoever. And our algorithm is able to discover images that look quite similar to the ones that we had as targets. They're not quite identical. You can see that there are differences. So if you start looking at the details, you can see that they're not quite identical. Here, there's a lot of blurring in this Chinese character. Our algorithm has problems with very distinct edges. Our Monet here is quite distorted, and so on. But it is quite remarkable that out of all possible images, our algorithm comes up with preferred tuning for these units in cases where we know the ground truth that is quite similar to what we would expect. So this algorithm can generate images in a vast stimulus space, and there are some limitations like smoothness, high contrast edges, and so on. So next we took units from a neural network, in this case again from AlexNet, where we know what the unit should respond to. And then we again try to discover, using our algorithm, what kind of images trigger high activation for those units. So let me just give you one example. We've done extensive testing, and I don't have time to do justice to all of this. I'm just going to give you one example. We look at one unit in the top layer of AlexNet, that is in the classification layer. This is the unit called unit number 599. This is the unit that's involved in classifying honeycombs. There's a misspelling here. Okay, so these are of all the images in ImageNet, that is out of about 1.5 million images. These are the 3 images on ImageNet that trigger the highest activation of these units. So the activation for one image is 27.6, etc., etc. So here in green, you see the distribution of activation values for all the images on ImageNet. So this is the probability density, this is activation, and the green here is all of ImageNet. So here, this point here corresponds to the maximum of all ImageNet images, and that's this picture here. So this is the image, the best image in ImageNet. Then we run our algorithm, we run Xdream, we start with noise, and at the beginning we start with images that show very poor activation, we gradually get better and better activation, and at the end we can generate images that are better than everything in ImageNet. This is the particular image that our algorithm generated here. If you squint enough, you can think that there may be some resemblance to honeycombs in here, but that's not the main point I want to make. The point I want to make is that we can generate images here that trigger much higher activation than any of the images in ImageNet, any of these natural images. So in a way, we can circumvent the problem of having to present 1.5 million images, just use our algorithm and very quickly come up with images that strongly drive these units in silico. Okay, so to summarize, we introduced this method called Xdream. This is largely unbiased, not completely unbiased, but largely unbiased. It works in real time, it's in a closed loop, and it can discover super stimuli, that is stimuli that trigger very high activation. It's not guaranteed to discover global optima, but it can discover global optima. It works, I didn't quite show this, but it works across layers, across different networks, across different developmental regimes. If you can go into all the details in the paper here. And it's also robust to many different changes in the algorithm itself. So we can change the search algorithm, we can change and use different image generators. We explored this quite extensively. I won't have time to do justice to all of that. For the aficionados, you can go into the paper for details. Okay. So, any questions so far?
Yeah, okay, I see that there's Suresh Krishna wrote in the chat, is high activation desirable for classification performance in the stimulus neighborhood?
Yeah, so that's a great question. So we're using high activation as the definition of what a neuron wants. It's not trivial that's necessarily better for classification. You could imagine that recognition and classification ultimately will depend on the population of neurons, and here we're only looking at a single neuron. So one possibility, and we're exploring that now, is that instead of maximizing the activity of a given neuron, we could try to maximize the activity of a population of neurons. And we can try to define what it means to maximize the activity of a population. And another possibility, as you sort of hint to here, we, instead of maximizing activation of a population, we might try to maximize discriminability, for example. We can maximize classification. So in principle, this algorithm should work with different loss functions, with different fitness functions. So here we're trying to maximize firing rates, but we could inject there any other loss function, including classification, and try to maximize that. We haven't quite done that yet.
I'd be happy to ask another question if it's possible. The starting point is basically some smeared or non-perfectly sharp natural images, as you said. But what if you would have started from just random noise? Do you think you would have you'd have reached the same results, or do you think it's worthless because these neurons were trained on world statistics, so they would not? I mean, that would have navigated you to things that are way out of the real world. I mean, these images that you do reach have some natural world statistics, so they are artificial, but they do resemble some real world. And now I'm just wondering.
Yeah, absolutely. So two comments on that. So first, I didn't quite show that, but you can go to the paper. We did explore different starting initial conditions. You can actually start with pure noise. It makes life harder for the algorithm, so it takes way longer to actually converge to real images. But it works too. So you can actually start with random images. Which images you start with can actually make a pretty big difference, and it can help with if you start with images that are already elicit high firing rates or high activation in the unit. Our algorithm is also was trained basically to generate images based on natural images. So that's partly why it makes it's much harder for the algorithm to generate Chinese characters or other kinds of images that look unnatural, basically. So in some sense, that's why I said it's not completely unbiased. Our algorithm is biased towards generating things that follow some of the basic statistics of the natural world, including correlations in pixel space, the power spectrum of the images, and so on. So if you want to generate images that are complete random noise or Chinese characters or things that look very unnatural, those things are generally harder for our algorithm to discover.
Thank you.
Okay. All right, I will go on unless there are any further questions. So I show you how this works in silico. Now I want to show you how this works in vivo. All of the work that I'm going to show has been done by all these talented people shown here. This is a monkey neurophysiology experiment. I'll show you only the recordings from the macaque monkey inferior temporal cortex, which is an area that we know to be quite critical for visual object recognition. The monkey is passively viewing images that are being flashed within the receptive field. Each picture is presented for 100 milliseconds. And we're going to do basically exactly the same thing that we just did with the computational models. We're going to present pictures. We're going to look at the firing rates, use those firing rates to drive the evolution of new pictures and continue the loop until convergence. So here we started by looking at the response of neurons to a set of 2,500 natural images. And here you can see the responses of these neurons. So these are neurons that respond more strongly to pictures of monkey faces and then to human faces and compared to other objects. For example, objects tend to trigger much lower activation for this particular neuron compared to all these other object categories from this set. And then we run our algorithm, we run the Xdream algorithm, and we can generate synthetic images. So this is the average response to all the natural images as a function of the generation number. So again, each loop, we call that a generation, each time we go through this loop. So in about an hour or so, the amount of time that we can hold the recording from the neuron and the monkey's attention, we can run about 200 or so generations. And here is the response of the neuron to all the synthetic images, to all the evolved images. So you can see, as we intended, that the firing rate increases from one generation to the next. not in a monotonic fashion. Sometimes the algorithm, goes in the wrong direction and corrects itself. And so on average, we're increasing the firing rate until we come up with images that trigger much higher activation than the natural images. So at the beginning, the early synthetic images, they're pretty bad. They trigger very low firing rates. And then at the end, our super stimuli, the images that we generate, can trigger much higher activation than any of these set of 2,500 natural images that we tested in this case. What do these images look like? This is a single example of a single neuron in macaque monkey inferior temporal cortex. Again, at the beginning, generation 0, we start with noise. This goes from left to right. This is generation 0, this is generation 14, then generation 15, and so on. At the end of this whole process, we're here. This is generation 209 for this neuron. And here we have the image that the algorithm came up with that triggers the highest activation for this macaque monkey inferior temporal cortex neuron.
Gabriel, may I ask a question? So this seems you're doing this like the series, the generation is sequential for a specific input, or do you interleave a few threads of this algorithm within each other? And would you see, like, if you test the sequence again, would you replicate the extent?
All great questions. So first, we interleave evolved images that is our synthetic images with natural images. So in between our synthetic images, we are presenting pictures of faces and houses and chairs and so on. That's how this picture was created. So it's the same neuron, and those natural images are interleaved with the evolved ones. As a minor note, we were very concerned about adaptation. We're bombarding the neuron with the same images over and over again. Indeed, I think adaptation is playing against us. We think that if we could sort of completely eliminate adaptation, we would actually get even higher firing rates. But despite adaptation, we can still increase the firing rate. The second question is about convergence, and I don't have the slide here. The short answer is sort of. So if you do the same experiment again, so that is you come again next week and you hope to be recording from the same neuron, and that's not trivial, but let's say that we think that we may be recording from the same neuron, you do the same experiment, you come up with images that are similar to this one, but not identical to this one. And so there's a couple of things there. So one is that we are not 100% sure that we're recording from the same neuron. And the second one is that we don't think that there's a single stimulus in the world that the neuron responds to. More likely, there's a whole manifold of stimuli. And there are many, many stimuli that probably trigger very similar firing rate. So we still need to figure out how to best study convergence. So the short answer is, yes, there's a significant amount of convergence, but the images don't seem to look identical if we repeat the same experiment. And we don't know whether that's variability in the neurons, whether that's because of this manifold idea, or sometimes we may just be recording from a nearby neuron, not the same neuron.
Okay, thank you.
I'm going to go through this very quickly. This is just a sort of a tangential comment. This is not what I wanted to focus on today. For the aficionados, there has been a lot of interest in creating computational models that can predict firing rates. Indeed, we can do that very well for the natural images. So we can take a computational model, we can predict firing rates. These are the real firing rates, these are the predicted firing rates. This works very well for all the natural images. This fails miserably when extrapolating to the evolved images. That is, these models do not generalize out of distribution whatsoever. I'm going to stop there and not comment anymore on this. I just wanted to make this comment for people who are interested in fitting firing rates, which is an exercise that many of us have been interested in doing. These models do not really generalize to these synthetic images. Okay, so this is just lots and lots of examples of doing the same thing. So here you see different neurons and the kind of images that were evolved by those neurons, together with the top 10 natural images. So these are the top 10 natural images that triggered activity in this particular neuron. This was the best evolved image. Here's another example. Here's another example, and so on. So in general, I think the notion of using anthropomorphic verbal descriptions for neural tuning seem to be insufficient. Many of these neurons seem to respond to pictures that are not easily described by English words. So if you look at the actual natural images, you may conclude that this is a face neuron. But if you look at the actual pictures that trigger higher activation, based on these kind of evolutions, you see that there are images that can trigger much higher activation than any of these natural images. I think that describing neuronal tuning based on simple English concepts is going to prove to be completely insufficient. Okay, so to summarize this part, we're proposing as the proof of principle an algorithm that can explore a pretty vast stimulus space. It's less biased than other approaches. It's not completely unbiased, but relatively unbiased, and it can work in real time, both in silico as well as in vivo. It can generate images that we refer to as super stimuli that can drive neurons at the same level or better than any of the natural stimuli that we have tested. This works in different visual areas. I only showed you a handful of examples from inferior temporal cortex. This has now been replicated in many different visual areas. The images have remarkable complexity. There's some degree of invariance to scale and rotation. I didn't quite show you that today. And they can approximately predict good natural stimuli based on the similarity. We can predict what are the preferred natural stimuli based on the similarity to this super stimuli. I didn't quite show you that point either. Okay, so now that we have this algorithm, and I showed you that it works in silico, it works in vivo, I want to use that as a proof of principle and focus on one particular example of a class of neurons, just to illustrate the complexities in studying neural coding and how I think that we should revisit how we describe neural coding in visual cortex. And for that purpose, I'm going to turn our attention to what are called the so-called phase neurons and phase patches in the macaque-monkey visual cortex. And I'm going to argue that phase neurons are not really semantic. They're not really encoding faces. They're encoding features that correlate with faces. In the same way that at the beginning, when I showed you a computational model that knew absolutely nothing about faces, you can still be deluded into thinking that you have phase neurons, even though there are units that were not tuned for faces at all, but correlate with faces. So to do that, first I want to define what I mean by semantic information. So for that, I want to use this picture here. So imagine that we have a neuron that's tuned to semantic information. For example, imagine we have a neuron that responds to the semantic concept of a fruit. So in principle, that neuron should respond very strongly to a picture of a lemon and also to a picture of watermelon here, despite the fact that these two images are quite distinct at the pixel level. Similarly, a neuron that responds to the semantic concept of tennis may respond similarly to a tennis ball or to a tennis court, even though these two pictures are as different as you can imagine in terms of their pixel similarity. So the vertical dimension here would correspond to semantic similarity. So these are concepts that we have based on English, based on usage, and so on. In contrast, neurons that respond based on physical similarity, they would show similar responses horizontally, especially at the top here. A lemon is very similar to a tennis ball. Even though semantically these are quite different, at the pixel level these are very similar. And so we would expect a neuron to respond very similar to these two if the neuron cares about physical similarity. I will argue that everything we know about ventral visual cortex suggests that neurons respond horizontally. That is, that neurons care about visual shape similarity, but not about this kind of semantic information. Semantic information must be there somewhere in the brain, and it would be fascinating to study that and to look for it. But in terms of what we know about ventral visual cortex and neural responses, I will argue that it's mostly physical similarity and not semantic similarity. It's very hard to study fruit neurons or tennis neurons, so I'm going to use the example of phase neurons that have been quite extensively studied. And I'm going to go through these rather quickly. We did a lot of different experiments, and I won't have time to do justice to all of them. So the basic idea is we're going to record from neurons in so-called phase patches in the macaque-monkey ventral visual cortex, and then we're going to generate, we're going to present natural faces and natural objects and natural stimuli. We're also going to use our algorithm to evolve images that trigger very high activation, and we're going to do behavioral experiments in humans to ask what those pictures look like. And when I say we, all of this work was done by Carlos Ponce, March Livingstone, Alex Bardon, and Will Shaw. We conducted 6 experiments. Most of them have the following flavor. We have a person fixate, we present a picture, and then we ask a question. So, for example, we ask people to describe the image in words. Or we do a 5-way categorization. We ask people, is this a face, a chair, a house, etc., etc. Or we force people in a 2-way categorization experiment. We ask, is it a face or not? Or we ask people to rate from 1 to 5 the degree of faceness of the image, 5 being very much face-like, 1 being not face at all. And then we also conducted 2 other experiments. One was a similarity experiment where we have one of our evolved images and we ask people to indicate which of 2 options it is most similar to. So is this picture most similar to the one on the left or on the right? For example, we could have a face on the left or and a non-face on the right, and we can ask then whether these evolved images look more like faces or not. And then another experiment we did was we show some of these pictures and then we ask people to click on the mouth. And so this is another way of assessing whether these pictures really look like faces or not. Okay, so I already told you that we can record activity of neurons in face patches. These neurons, according to traditional definitions, they respond more strongly to faces compared to non-faces. So I won't show you the results from all the experiments. I'm just going to show you a few of those experiments. So the first experiment I want to show is the one where we ask people to click on the mouth. So here's a picture of a real face. And lo and behold, when we ask people to click on the mouth, this is what they do. So it's not a very complicated task. It's pretty easy. Most of the clicks really end up on this woman's mouth, as you might expect. When we ask people to click on the mouth and we present the chair, these were actually literally the instructions we gave. We said, click on the mouth. Of course, that doesn't make any sense whatsoever in the context of a chair. And this is what people do, okay? So people still click on the center because perhaps there's a bias towards the center, but they also click sort of everywhere, okay? So there is much more random clicking. And the critical question for us is when we show our evolved images, and again, these are images that trigger very high firing rates in these so-called face patches. In many cases, many of these images trigger higher firing rates than pictures of actual faces. We asked people to click on the mouth, and this is what they do. These are the locations. These red dots indicate the locations of where they click. And we do that for images generated from neurons within face patches, as well as images generated from neurons outside face patches. So to try to put a number to these, we calculated the entropy of the clicks. So the entropy of the clicks essentially is a measure of how much variability there is in the position of these clicks. So if this number is very low, that means that there's a lot of clustering in the clicks. If this number is very high, that means that there's a lot of variability. So when we present pictures of face photographs, we see that there is relatively low entropy. So this corresponds to this example here. So that is, people are quite consistent in general in terms of where they click and when we ask them to click on the mouth. In contrast, when we have non-face photographs, such as the chair here, there's a much wider range of entropy. That is, people click on lots of different places. Sometimes there is low entropy, and that may indicate cases like bias in the way in which people click for some of those images. But overall, there's a very clear distinction between non-face photos and face photos in how people respond to this strange query of clicking on the mouth. Critically, when we look at images that are evolved from face neurons, we see that there's a much higher entropy, almost the same entropy as what we see for the non-face photos. And the same thing happens when we have images which are evolved from non-face neurons. As a minor technical point, the images that we generate are somewhat distorted, as I showed you. So we don't generate perfectly knitted pictures with strong edges. So we generated images that really look like faces, but have the same kind of distortion, and we call those stylized face photos. So these are evolved images where we know that the ground truth should be a face, as I showed you before, and we can generate images that really look like faces, but they're slightly distorted. The edges are a little bit blurred, and here we see that the entropy increases a little bit, but it's still much lower than what we get for evolved images. So in other words, for these images that are evolved from so-called face neurons, or evolved from neurons in face patches, people are quite dispersed in where they think the mouth is. So I want to show you quickly two other experiments. So in this experiment, this was a two alternative forced choice binary discrimination. We asked people, does this picture contain a face, yes or no? Okay, so we show a picture like this one. Is there a face? People are supposed to say yes. We show a chair. People are supposed to say no. And so when we show pictures of face photos, essentially everybody responds, yes, this picture contains a face. When we show pictures of non-face photos, these pictures, most people say, no, these images do not contain a face. And when we show pictures of evolved, generated from evolved neurons, I don't know how to get rid of these. Oh, there it is. Okay, so when we show these images that are evolved from face neurons, by and large, we see that people do not consider these kind of images to actually represent faces. Intriguingly, for me at least, when we show images evolved from non-face neurons, there's only a small decrement in how much people consider these to be faces. So the images that are generated from face neurons are only slightly more face-like than those images generated outside these so-called face patches. Here's another experiment. We thought that maybe this binary choice was too restricting. So instead of asking people, is this a face, yes or no, we asked the question in a slightly different way. We asked people to rate from 1 to 5, how much do you think that this contains a face? And so here again, the face neurons, the face photographs trigger responses that are pretty close to 5, that's the maximum, whereas the images evolved from face neurons ended up yielding pretty low ratings, slightly better than, but not much higher than those images evolved from non-face neurons. Okay, so to summarize this part, evolved superstimuli from face patches are labeled by humans as distinct from face photos. And here I should point to a very strong caveat of this study, which is that we're studying neurons from monkeys and we're asking for behavior in humans. So it's conceivable that monkeys perceive these images in a completely different way. We think that there are a lot of similarities between how monkeys see pictures and humans do, but we're studying behavior in humans with neurons from monkeys. We hope that somebody will be able to do the experiment of recording the activity of neurons in humans, do this experiment and test humans, conversely study behavior in monkeys. But we're studying behavior and neurons in different species here. There are minimal differences between neurons inside and outside face patches. And we argue that the so-called face neurons are better described as tuned to visual features, that is, shapes, but not the semantic category of faces. So we don't think that the word face is an adequate description of what these neurons respond to. They respond to complex features that happen to be correlated with faces, in the same way that we could get presumably face neurons in a network that did not have face neurons at all. In a network that responds to pictures of biological cells and eyes and roses and so on, we could discover units that respond more to faces than non-faces. Here we can also discover neurons that respond more to face photographs, even though those neurons are actually tuned to much more complex shapes that are not well described by the word faces. Okay, so to summarize and to finish, I have a horrible slide at the end, which has a lot of text. I just wanted to go through a lot of speculation, technical comments, disclaimers, and caveats to everything I told you today. Okay, so first of all, I want to say that superstimuli may represent local optima. I showed you that in some cases we can find the global optima, but in general, we are not guaranteed, and we might very well be stuck in a local optimum. So somebody come back with a better algorithm in the future and show that they can elicit, they can generate images that elicit twice the firing rate that we have. There's a limit that's given by the refractory period, so you cannot go to infinity in terms of firing rate. There's a physical limit to how fast neurons can fire, but we haven't reached that limit. So it's conceivable that there are better pictures than our superstimuli. We don't think that there's a single picture. We're not arguing for a grandmother cell-like representation. I don't think that there's a single picture that a neuron likes. More likely, there's a whole manifold of superstimuli. And in fact, if you look at our evolutions, there are lots and lots of images that are related, that they're similar, that trigger very high firing rates. So it's better to describe this as a manifold of stimuli rather than a single superstimulus. The particular stimulus may very well depend on the task, the spatial context, the temporal context, development, experience, dynamics, all kinds of variables that we did not explore. All we did here was passively show pictures to a monkey and look at the tuning preferences. But presumably, there are lots and lots of things that could change that type of tuning. We haven't quite explored that yet, including adaptation, including the temporal context, including the training regime of the monkey, including the task, and so on. We showed one particular metric, that is, we tried to increase firing rates, and we use that as our basic definition of neural coding, but that's not the only way to think about neural coding. So we can, in principle, use Xdreams to maximize local field potential in different frequency bands. Someone asked, and we can look at different population metrics. We can try to maximize spike synchrony. In principle, any well-defined mathematical property, one could be able, one could try to generate stimuli that will maximize that particular neural code metric. In general, showing that the response to a particular picture or a particular class of pictures A is better than B does not imply any semantic information. So we and many other people have done experiments where we show pictures of faces and houses or chairs and tables and so on. The fact that the neuron will respond more to one than the other does not imply that there's any semantic information there. There has to be semantic information somewhere in the brain. We use language, we use these semantic categories continuously. We don't think that there's any evidence yet that semantic information is stored within the type of ventral visual cortex neurons that we and other people have studied. I would contend, and this is perhaps a polemic and issue that many people will not agree with, that it's time to abandon anthropomorphic description of neural tuning. So I'd like to abandon the notion of face neurons, chairs, place cells, curvatures, and so on. And I think we really need computational models to describe the kind of features that neurons are tuned to, which may not have a one-to-one mapping onto English words. So I would like to argue that ventral visual cortex responds to complex shapes, which are partly described by current computational models, but I also showed very quickly in one slide that our current computational models are quite bad at extrapolating to out of distribution images. But I think this is the right direction to go. I think we need better computational models to describe the neural coding properties of neurons in visual information. Despite the fact that these neurons are not tuned to chairs and faces and natural objects, we can still decode natural information from this shape-tuned tuning. That is, you can have a dictionary of features where the units are not responding to any one particular semantic category, and you can still discriminate chairs from tables. You can still discriminate faces from houses. You can still identify one face from another just because you have a sufficiently rich dictionary of very complex features. That is, we don't need chair neurons to recognize chairs. We just need a lot of visual features that are sufficient to discriminate a chair from a table and so on. And finally, I want to say that we have focused on vision. We think that vision is the most interesting thing to study in the world. However, this can be applied to many other domains. So in principle, one could think about recording from auditory cortex and following the same approach, starting with noise and coming up with, according to this evolutionary algorithm, coming up with particular measures or music or sounds that will trigger very high activation in those neurons. One could in principle apply this to other cognitive functions, as long as there's a way to generate stimuli. And as long as there is a metric to evaluate those stimuli. In our case, we can generate images by using image generators, and we can measure firing rates. As long as we have those two key ingredients, then in principle, this can be applied to many other problems. In the context of vision, I think that that's particularly easy because we know how to generate images. In the context of auditory stimuli, maybe also. In the context of high-level cognitive function, that seems much, much harder. So if we study neurons in frontal cortex, for example, we don't know how to generate cognitive tasks or complex cognitive stimuli to trigger activation in those neurons. But in principle, at least, if we have a good generator and we have a good metric, in principle, this could be extended to other cases. Okay, so this is a very bad, very crowded slide, but I just wanted to preempt a lot of comments, caveats, and questions. I'll be happy to take questions. Again, I'm sorry I'll have to go in about 5 minutes, but I'll be very happy to take questions and otherwise continue via email.
Gabriel, thank you very, very much. So first of all, I'd like to ask everybody to unmute yourself, and let's give Gabriel a big thank you for a wonderful and really exciting talk. And I myself have a question. If you think then that the face patches, following your logic, are not really face patches, I mean, maybe if everything is kind of like distributed, maybe what are the borders, or are there any areas at all other than cytoarchitectural borders within high order visual cortex? It's just a thought.
Yeah, I completely agree. So I think there are borders that are imposed by the gyra and sulci in the brain. So at some point in the brain that everything is folded. Those are real anatomical borders. Other than that, I am less sure about borders. I think that there's a continuum. Everything we've done and other people have done suggests that nearby neurons seem to have very similar tuning preferences. It's a little bit like asking where it rains and where it stops raining, right? If you keep walking, let's say that it's raining in Tel Aviv, and you start walking towards Haifa, at some point it will stop raining, but you say, well, where exactly does it stop raining? Well, it's not a single point in space, right? So yeah. I think similarly, you'll have neurons that are tuned to some stimuli. You look at the nearby neuron and it has similar preferences, and so on. But I don't think that there's a real sharp border. At least I don't know any evidence of a sharp border, except of course for the anatomical ones, right? So at some point you land into white matter, you land into
the work by Kalanit and the collaborators, where they found the cytoarchitectonic borders within the fusiform, for example, and stuff like that.
Right, absolutely. So I think those may be real borders, but in general, in terms of tuning, I think it's much more of a continuum than real major borders. That's my opinion. Again, all of this requires much further scrutiny.
Thank you. Fascinating work. If you've got another question, Ben wants to ask one, but I know you are short in time. So Ben, maybe quick question.
Thank you. Really fascinating stuff. My question is, do you think that we may have, or the monkeys may have, tuning for these particular abstract features and not others? Because on a population level, they may be helpful to differentiate between what we would describe as semantic categories and within them.
Yeah, absolutely. I think of this a little bit as, we have a handful of letters, and with those letters we can form essentially infinite words. So in a similar way, we have a lot of complex features, and by combining those features, we can discriminate different faces and different Chinese characters, and different chairs and tables and so on. So ultimately, I think behavior perception will depend on the population. As long as you have a sufficiently rich dictionary of features, you don't need chair neurons to be able to recognize chairs. With sufficient number of complex features, some that respond to curved lines, others to 45-degree lines, others to lots of color and so on. By combining all of these, you put 1,000 of these features together, and you can very easily tell apart a chair from a table, one chair from another chair, your chair from somebody else's, all kinds of distinctions, as long as your dictionary is rich enough. So I guess I'm just restating what you just said. But
Thank you.
Thank you very much. So, Gabriel, we're actually, I mean, you were supposed to be somewhere else. Are you going to teleport yourself? Just wondering. Do you have
yes,
time?
I'm going to teleport myself. I really apologize because I love. Yeah, so once more, I really apologize because I love discussions and questions. So please feel free to email me if you have further questions and I didn't get to answer your question. So please don't be shy and send me your questions. Unfortunately, I do have to go to a thesis defense now. So
Thank you very much. It's been a pleasure having you. Wonderful work. And I want to tell everybody